10.2, scientists investigated the function of eukaryotic cell protein called cyclin A. This protein is thought to be involved in the binding of the enzymes required to start DNA replication. So C, cells were untreated. D, added an antibody that binds to cyclin A. E, added RNA that prevents translation of cyclin A. And F, added RNA that prevents translation of cyclin A and added cyclin A protein. And we are given the results in table 5. We can see that 91% of the cells were still dividing in the control. In D and E, this reduced rapidly, and F, it was around about the same. So the question is asking us to explain these results. So suggest an explanation for the results in table 5. So we're looking to suggest an explanation as to why in D and E there was a dramatic reduction in the number of cells that were replicating. But in D, they added an antibody that binds specifically to cyclin A. So if the antibody is binding to cyclin A, then cyclin A's job is to start replication of DNA. So if the antibody is binding to cyclin A, then therefore the cyclin A can no longer bind with the enzyme or the DNA to stimulate the DNA replication. Therefore, the number of cells that were replicating will be reduced. For E, they had RNA that prevents the translation of cyclin A. So that RNA is interfering with the translation of cyclin A, which means that the cyclin A is not going to be translated, so therefore it cannot then bind with the enzyme or the DNA to initiate the cell replication. For F, although they had the RNA that prevents the translation of cyclin A, they also added the cyclin A protein, which then would have been able to bind with the enzyme in order to cause the DNA replication. So therefore, on our mark scheme, marking point 1 is recognising that with treatment D, the cyclin A cannot bind to the DNA or bind to the enzyme, so it can't initiate DNA replication because the antibody has bound to it. For marking point 2, the idea with treatment E, the RNA interferes with the mRNA or the tRNA or the ribosome or the polypeptide formation, because it is interfering with the translation of cyclin A. In treatment F, they added cyclin A, which then could bind to the DNA or the enzyme to initiate DNA replication. Or you could say that treatment F shows that it is the cyclin A that's being affected in the other treatments. Or treatment F shows that cyclin A allows the enzyme to bind to the DNA. Or some of the cells in D and E can continue with DNA replication because they have a different cyclin allele. Or some cells in D or E can continue with DNA replication because the antibody has not bound to the cyclin A. Or some cells in E can continue with DNA replication because they contain previously translated cyclin A.